So, good afternoon. Do you ever feel like there's not enough hours in the day? Right, so that's definitely how I've felt lately, and I'm sure a lot of you feel a lot, but you know, it's getting to be crunch time here. Get everybody in that must be in. Okay. All right. So I do appreciate you coming. I know it's easy to forget there are 125 people in this class, only about 30 of you come every time. I do appreciate you coming, and I think it's good for you too to be here. As of like a few minutes ago, I have everybody, I think, that taken the test, that is going to take the test. A few people I haven't heard from. I'm going to assume a week later that you're not going to take it. So these were the 5 most commonly missed questions. These were the 5 questions that less than half of you got right. So the only 5. So this one, I think only 38% of students got this one correct. Membrane attack complexes. So remember, this is when the C6, C7, C8, or 5, C5b, C6, C7, C8, and lots of C9s come together to poke holes in membranes. All right, the correct answer here, this is going to be especially lethal to gram-negative cells. Gram-positive bacteria, the peptidoglycan layer is too thick and it doesn't poke through. So with gram-negatives, goes through the outer membrane, peptidoglycan, and the plasma membrane. All right, so we talked about that a little bit. This one, probably most of you knew the mechanism of action of SXT. So I asked the so what? It interferes with enzymes to synthesize folic acid, which is necessary for the synthesis of DNA. All right, we need that in our cells as well. We get ours from the food we eat. Bacteria have to make their own. All right. All of the following are possible benefits of fever. Okay, except, okay, so all of them are right except one. There are a good number of questions sort of inadvertently on the final that say all these are true except the following. And I know when you get too many of those, it gets complicated, but just make sure you take the time to read the questions carefully. This one is not true. Fever, as far as I know, does not increase the production of membrane attack complexes. If you can find that in the literature somewhere that's a thing, then I would give you credit for that. But as far as I know, I just made that up. Okay, the other ones are true. We talked about. And there was 3 methods that complement is activated. In that lectin pathway, lectins are what we make in the liver, and mannose is a sugar on the surface of pathogens. All right, so the last one was the correct answer there. All right, last one, activated macrophages. Which is true of activated macrophages? The temptation was there that they recognize a wider variety of PAMPs, pathogen-associated molecular patterns. There's only 10 or 11 PAMPs that we know of, so that's not the case. Activated macrophages are larger than ones that are not activated. That was the correct answer there. All right, so be sure to stop in and see me if you want to look over the exam. I do have office hours all morning tomorrow, and I can stay till about 2 or maybe even 2:30 tomorrow. I'm thinking about having office hours on Friday morning. So if that would be helpful to any of you, let me know and I'll definitely do that. So send me an email, hey, can we meet at 10 o'clock or whatever. I have so much to do that I'll be more efficient if I come into campus and do that. So if that would be helpful, send me an email, let me know that you're coming so I can make sure that I am here, or we can Zoom too, whatever you prefer. But if you want to Zoom and you want to look at your exam, then I have to be in my office because I don't take those exams home with me. All right, so where we left off last time, we were talking about anthrax. So remember Bacillus anthracis, that's a gram-positive rod-shaped organism. So we're still in chapter 19, which is pathogenic gram-positive bacteria. And the bacillus are mostly a genera of organisms that are not pathogenic. They're just soil organisms for the most part. There are a couple exceptions. The big one is Bacillus anthracis. And how the disease manifests itself depends on how the patient is exposed to the spores. All right, so cutaneous anthrax is if you get the spores in a wound. That's the kind that's not as serious, potentially can kill you, but not likely to. Pulmonary anthrax is when you breathe in the spores. So we finished up last time talking about pulmonary anthrax. That's the form of anthrax that killed 5 people when spores were mailed to Congress in 9/11 in 2001. Okay, but remember, anthrax is typically associated with farm animals. All right, so people in close association with farm animals, it's considered zoonotic. All right, the third way that we can contract anthrax is if we ingest or swallow the spores. All right, so that's called gastrointestinal anthrax. And this is typically how animals are going to get anthrax. So they're eating off the ground, or they're, the spores are in the soil, so they're eating the grass or whatever off the ground, and typically they're going to swallow it. All right, so this is going to cause necrosis. Remember, that's just death of tissues in the gastrointestinal system, hemorrhaging of intestines. Sores in the mouth are common, as well as severe ascites. Okay, that means kind of an abnormal swelling, abdominal swelling. And I tried to find a good picture of that and wasn't successful. Okay, but that ascites or ascites there, abdominal swelling. It's not typically an infection, though, that kind of lasts very long. Sometimes animals will be out grazing in the field one day and the next day they're dead of anthrax. So it's typically a pretty fast acting disease, near 100% mortality if it's not treated. And even when it is treated, it's very likely to kill you. Okay, just like pulmonary anthrax. All right. So how do we treat anthrax? To be honest, what antibiotics would be used might not be these exact antibiotics, but things like penicillin or tetracycline have been used in the past. If someone is thought that they perhaps have been exposed to anthrax, but they're not showing any symptoms yet, they'll give ciprofloxacin, which is a fluoroquinoline, just prophylactically. So even though they're not showing symptoms, just in case, because it's such a serious infection, might be given antibiotics to ward off the infection. Okay, and approved in 2012 were some monoclonal antibodies to try to neutralize the toxins. So really, in terms of virulence factors, there's a couple of toxin, edema toxin, lethal toxin. Again, if you can kill the organism, but when the toxins are still circulating, then that's really what's typically causing the disease. So I'm not, I don't think, we have so few cases of anthrax normally, that I'm not sure, how effective that is, whether that's been tried in other countries where it might be more prevalent. So I don't really have a good feel for that. Okay, what about a vaccine? So, treatment's not always effective. Do we have a good vaccine? Well, we have a really good vaccine in animals. And about 120, 130, 40 years ago, Louis Pasteur, this was one of the early vaccines that was developed. It's a really interesting story to kind of read about how he did that. But anthrax was a huge scourge for farmers because it would not only, kill the animals, obviously, which is a financial hit, but then the farmer couldn't graze other animals in the field because the spores were in the field then. So it was a huge problem, and he found an attenuated vaccine that worked well in animals at that time. I'm not really sure what form of vaccine it is, to be honest. I should look that up. We do have a human vaccine for anthrax, but it's not a very good one. You have to take 5 doses over about an 18-month period, and about 1% of the people that take the vaccine have really severe reactions to it. And not just, you have a little fever or a little pain, or like neurological reactions. So for that reason, it's not something that we widespread give to everybody just in case of a terrorist attack. If you're in the military, you probably need to get it, especially if you're traveling overseas somewhere like the Middle East, where the spores are more prevalent in the soil. If you work with anthrax in the lab, then you might get it, but and the protection only lasts for a year, and then you have to have a booster again. So it's not a great vaccine. Okay. Questions about anthrax? Anything? All right, so a closely related genera to Bacillus is Clostridium. All right, this might look a little familiar. And if you're in lab this week, we did a spore stain this week with a Clostridium sporogenes. Okay, so these are also gram-positive rod-shaped organisms, and they're pretty big rods. So like this picture right here is not a gram stain. I think it was just like with some India ink or something. They're pretty large rods. Where do we find Clostridium? Kind of in the same places that we might find Bacillus, in the soil, vegetative debris. Like Bacillus, they're considered saprobes. So remember, that means that they feed on basically dead organic material. A lot of fungi are saprobes. So what they have in common with Bacillus is that they are the other genera that are kind of the famous endospore producers. Okay, so big gram-positive rods, you can see those in this picture here. They're not taking up the dye here, so this is not a spore stain. Okay, and the other kind of distinct thing about Clostridium is that they are mostly anaerobes. Okay, so some of them are strict anaerobes, some of them are aerotolerant anaerobes, but most of them lack the ability to detoxify those toxic oxygen forms, and that's what makes them anaerobic. All right, so they're not going to respire aerobically. All right, so some examples of Clostridium. There's some really interesting organisms because they cause some really nasty infections because they produce, it's the toxin production again. So there's a couple of organisms here that produce pretty potent neurotoxins, Clostridium tetani and Clostridium botulinum. It cause tetanus, botulism. Both of those organisms, again, produce a neurotoxin that either won't let our muscles relax in the case of tetanus, or won't let our muscles contract in the case of botulism. So both potentially deadly infections. Perfringens is sometimes causing food poisoning, but it is also the cause of gas gangrene, which just means, gangrene just means death of tissue. So you're going to get some kind of a deep wound, and that, again, just can cause the tissue death. It's supposed to smell really amazingly awful. But we're not going to have time to really get into those 3 organisms, so don't need to worry about those. We're going to talk about C. diff here just a little bit because you're a lot more likely to come in contact with that at some point. All right, so Clostridium difficile, the infection is basically the same as the organism. We call it C. diff for short. This organism is maybe part of our normal microbiota. And I was reading about that. Well, how many people have this as part of their microbiome? Apparently, it's more common for children to have it in their large intestine, and we're more likely to lose it as we get older, which is sort of not what you would think, because you think more likely are older people getting C. diff infections, but potentially you could have a little bit of C. diff in your large intestine. It's not causing any problem. You have it there in low numbers. We do consider it an opportunistic pathogen because typically someone would have problems with C. diff if they go on a course of strong antibiotics, or maybe several courses of strong antibiotics that wipes out the variety of microbes in their large intestine, and the C. diff takes over. All right, so opportunistic pathogen, we'll say when the normal microbiota is altered by antibiotics. Okay, so you're really, really unlikely to get C. diff unless you've been on antibiotics. It's not unheard of, but usually it's when somebody's on antibiotics. So what's the infection look like? It causes a really persistent diarrhea. So one that doesn't go away. So it really contributes to weakening individuals. Maybe doesn't kill people outright very often, but definitely contributes to mortality. We think there's something like 3 million cases in the US every year of C. diff just in hospitals. And then people get it in the community as well. Okay, so it can, in addition to diarrhea, cause what we would call pseudomembranous colitis. And that's just a little bit more serious ulceration of that lining of the large intestine of the colon, or the cells lining the large intestine. Okay, and that even organism can potentially perforate the large intestine as well. So that's a huge problem when you get the microbes that were in your large intestine now spilling out. That's potentially deadly situation. So very serious diarrhea that's hard to get rid of. Virulence factors, we won't mention a whole lot here. There's a couple of toxins that the organism produces, and toxin A is an enterotoxin. Remember, that's a toxin that kind of targets your small and especially large intestine to make that lining a little bit more leaky. Toxin B is a cytotoxin. That just means a toxin that causes death of cells. So together, those 2 toxins will cause the death of cells, we'll say epithelial cells, lining the colon. And really, in addition to that, we could add fimbriae in there. Remember, fimbriae are kind of like flagella, only they're a lot shorter, and they're important for attachment. Okay, so the C. diff can attach to the correct cells in the large intestine instead of getting kind of washed out. Okay, so how does one get C. diff? Like I mentioned, it's usually, if either it's part of your normal microbiota and then it has a chance to take over. You can also be exposed to the spores. Okay, so I mentioned last time that typically organisms that sporulate would only do so in the environment. So it's a survival feature, right? So when conditions get harsh, they sporulate until conditions improve. here's the notable exception. C. diff will sporulate in the host, okay, in our bodies, and then they actually, the spores are released in the feces. Okay, so if you have worked in a nursing home or hospital and have been charged with cleaning up some C. diff diarrhea, you would have had to gown up. You know, this was even before PPE was in fashion. You would have totally gowned up, you know, rubber gloves, everything, in order to clean it up. That's to protect you, right? So you don't accidentally, you know, get the spores, you know, rub it in your mouth or something like that. Even more importantly, you know, assuming that you're healthy, you're really not very likely to contract C. diff just by, you know, being exposed to spores. Even more importantly, so you don't carry them to the next room where that patient's on antibiotics and is likely, you know, be much more susceptible to C. diff. So, all right, so can be transmitted in the diarrhea, and that C. diff diarrhea is supposed to have a particularly pungent odor. So we mentioned this before. C. diff is doing all these different kinds of fermentations that have particularly nasty smelling fermentation products. So the whiff of the diff. All right, so what do we do about it? How can we treat C. diff? The first course of action would be to put the patient on antibiotics, and typically vancomycin is the first choice there. That's an antibiotic where typically is only given by IV, but in order to treat C. diff, we have to get the antibiotic into the digestive system where it's going to work. So in this case, this is the only case where you would take vancomycin orally. And if that works, then that's great. Unfortunately, vancomycin doesn't always work. A lot of C. diff has a lot of resistance to antibiotics. So very often, especially if, you know, maybe the patient's a little immunocompromised or in frail health anyway, antibiotics don't work. And you might try a couple of courses of antibiotics. If that diarrhea is persisting, there is an option now that's been around for a few years. It's getting more popular. We used to call it fecal transplantation. I looked up this week, and what they're calling it now is FMT. Okay, so that stands for fecal microbiota transplantation. Okay, so it makes sense. How do you repopulate the large intestine with kind of a normal variety of microbes so you can outcompete that C. diff again? Well, you put in a little bit of feces from somebody that's healthy. All right, so they used to do this with an endoscope. They would get maybe a relative of the donor, screen them really well for any kind of infectious disease that they can think of. And, you know, would use an endoscope and put a little feces in there. They are getting better at this. We can now deliver that feces by pill. So it definitely, you have to get over the ick factor, but this has around a 95% cure rate, which is much better than the antibiotics. All right, so a few years ago, the only kind of capsules that would kind of dissolve at the right time, meaning they'd pass all the way through the stomach and not dissolve till they were in the large intestine, were clear capsules. All right, so, and they're big, they hold like 2.5 grams. So again, it's the ick factor, but patients would have to swallow 10 of these capsules a day for 3 days. All right, so they found that a huge dose is going to be more effective than maybe just taking a little bit. All right, now they have some capsules now that don't look quite so nasty, that are colored, but you do have to freeze them because the microbes are alive, and right, you want to make sure that they're alive when you take them. Okay, so to me, this would be a pretty sweet job. Maybe some of you donate plasma for a little extra money. You know, you got to go and be hooked up, sit there for a few hours. You'd be a fecal donor. That seems like that would be pretty easy. So if you live in places like this picture came from a company in Portland, Oregon, you know, and they would screen you real good, make sure you're healthy, and then, you could make some cash that way. Seems like a pretty sweet gig. The one problem with this is a couple of years ago, somebody died. They got a fecal transplant, and they found that he had a particularly aggressive multi-drug resistant strain of E. coli. And then somebody else got sick with that same strain, and they both had fecal transplants, and they traced it back to the donor and found that strain of multi-drug resistant E. coli that he was carrying that. So we have more to learn about what kind of things we need to screen for. They were screening for different pathogens, but just not that particular one. So that's kind of a setback. This is still considered a, what do you call it? What's the word I'm looking for? Exploratory. It's not approved by the FDA yet. So we do about 10,000 of these a year, but maybe at some point it will become a little bit more common. We just got to work out all the kinks to that. So, all right, questions? Anything about C. diff or anthrax? All right. All right, so we are still in chapter 19. Seems like we've been in chapter 19 a while. Okay, so I want to talk a little bit about Mycobacterium and tuberculosis. Okay, so how did I choose, which pathogens to talk about? There's a whole lot more of ones in the book. TB is a good example of a infection that we thought was going to be eradicated, but that has not been the case. And it's also, if you'll remember, an organism that we consider acid fast. Okay, so they have this organism, this genus grouped in the chapter with the gram-positive organisms. It really, most of the time, it's considered not gram-positive or gram-negative, but we put it in its own group, has this kind of cell wall that we call an acid-fast cell wall. Okay, so they threw it in with the gram-positives because it doesn't have an outer membrane, but really, we kind of consider it its own group. And there are, about 75 different species known of Mycobacterium. They infect, various animals and cause, various infections in humans that are pretty rare for the most part. The ones that are most well known, Mycobacterium leprae, that's the causative agent of leprosy. So an ancient scourge of mankind that's been around a long time. It's a disease of nerve endings. This organism actually has an ideal temperature of about 30 degrees Celsius. So it likes the extremities. So fingers, nose, toes. Okay, and over time causes these deformities, weakens the patient. Treatable with antibiotics, especially when you get it early on. There is a Mycobacterium bovis that causes a tuberculosis, a bovine tuberculosis, so tuberculosis in cows, deer, elk, buffalo, things like that. They can pass that on to humans when we drink their milk. So when we drink cow's milk, we can get bovine TB. It's kind of a milder form of tuberculosis. Now that we pasteurize our milk, that's basically unheard of, at least in this country. But in other countries, developing countries where they don't pasteurize their milk, that's still kind of a big thing. All right, so we're going to spend most of our time on here, Mycobacteria tuberculosis. Of course, this is the causative agent of TB in humans. And I think it was 1882 that Robert Koch discovered the causative agent for tuberculosis. Up until that time, we thought that TB was a cancer because it was slow growing. It tended to run in families. It's interesting. We set up these, sanatoriums all over the world, mostly like at high altitude. We thought that was good for TB patients to breathe the clean mountain air, and they advertised for patients to come and breathe the clean air. Well, they didn't know it was infectious. They knew it was infectious, and they might not have wanted to get people to come there. But at that time in 1882, about 1 out of every 7 deaths was due to TB. So extremely successful pathogen. All right. And when we developed the first antibiotic, 1946, I think, was when streptomycin came out. We thought, okay, we've got TB licked now. We've got a drug that works against this. The American Lung Association switched its focus from TB to another thing that damages our lungs, smoking, because they thought, okay, TB, we're not going to have to worry about that anymore. Well, that's not happened. Today, somewhere between 1 out of 3 and 1 out of 4 people worldwide are infected with tuberculosis. Right. So obviously, it has not gone away, especially sub-Saharan Africa. Here, we have a really high incidence of TB. Southeast Asia as well. It's a huge problem, especially when people are HIV positive. HIV and TB go together really closely. All right, so incidence is low in the US, but with people traveling all over the world, and we could be exposed to it. So it's something that we need to kind of know how it works. All right, and this is a little bit misleading, because really only about 10% of the people that have TB probably have active TB. So it's a complicated disease. It can go latent. Yes, question. Okay. So why do TB and HIV go together? Well, especially before we had good drugs to treat HIV, or people in areas of the world that don't have access to those drugs, because HIV knocks the immune system down. TB just takes hold. Okay. People that are healthy are less likely to contract TB when they're exposed to it. Okay. So, yep. Thanks for asking that. Okay. So again, it's complicated infection. We're going to kind of hit the basics of that. But it can be latent and may not actually activate. All right, this was just some data that I didn't bother to post, but now in the US, we have somewhere, on the average of about 10,000 cases of active TB. I looked on the CDC website right before I came to class, and the CDC estimates that about 13 million people in the US have latent TB. So that could be people that don't even know that they could potentially activate into TB. So it's a little bit more widespread than what we might think. But the incidence is going down. Here, the height here was early 90s. That was kind of the heights of the AIDS crisis. So again, as we've gotten better at treating HIV, that's kind of improved our outlook with TB as well. All right, so let's take a look at kind of the basics of the path of infection of the Mycobacterium tuberculosis. Okay, so although TB can infect almost every area of your body, it almost always starts in the lungs. Okay, so a patient inhales it, and then it gets into the macrophages. You have lots of macrophages in your alveoli. Okay, and so although TB can infect different kinds of cells, it seems to particularly like to infect those macrophages. And of course, that's a cell that's long-lived, that's traveling throughout the body, so it will eventually disseminate. And of course, when those macrophages are dying, then they're not presenting antigen and calling into that adaptive immune system. So that's a problem. So the organism replicates freely in the macrophages. We'll talk a little bit about the virulence factors that enable it to survive inside macrophages instead of getting digested. And when the patient first encounters it, they're going to have flu-like symptoms for maybe a week or two. Okay, so at that point, some people will fight off the infection. Okay, their immune system will get a hold of it and they'll clear it. Okay, so especially if you're well nourished and you're healthy to begin with, you're going to clear it. Somebody that's HIV positive is probably not going to be able to clear that. Okay, for some people though, I don't know what percentage, instead of clearing it, your immune system comes to kind of a stalemate with the mycobacterium instead. Okay, so it forms these, what we call tubercles in the lungs. Okay, so a tubercle is kind of a specialized name for a granuloma. A granuloma is kind of an abnormal aggregate of cells. And you can get granulomas with other infections, but like I said, this is really common for TB, so we have a special name for these granulomas. And basically what happens, you have some live cells in the middle here, but then it's kind of surrounded by lots of dead cells. Collagen is deposited around there. So we've got this kind of mass of cells, some alive, some dead, that's kind of contained, right? So when those live organisms are contained, then the patient is not infectious. Okay, so we would call this, when a patient has these tubercles, we would call this a latent infection. Okay, or you could also refer, I mean, primary TB can refer to, you know, when you just got those flu-like symptoms and you clear it. It can also refer to a latent infection. So you've been a little sick, but your body hasn't totally cleared it. Okay, and those tubercles can just sit there basically until the end of a patient's life, indefinitely. For some reason, in about 10% of patients with latent TB, they will eventually reactivate. I have that written on the next slide. Typically, when it reactivates, when will it do that? Maybe if the patient becomes immunocompromised for some reason, they become HIV positive, maybe they get cancer, maybe they're just really old. But we can get kind of an abnormal necrosis called Occasious necrosis. Apparently there's like 7 different kinds of necrosis. Okay, so this Occasious necrosis, this refers to it kind of a cheese-like consistency. Okay, so basically the inside of this tubercle, you get, you know, changes that, to be honest, I don't totally understand the mechanism there, but we get some kind of pus formation, and the result is that live bacteria are spilling into the lung from that tubercle. Okay, so at this point now, we would consider it secondary TB or reactivated TB. Okay, so we've got these live organisms inside that tubercle that are now leaving the tubercle, spilling out. Okay, so again, only about 10% of people progress to this that have those tubercles, but now the patient is infectious. Okay, so the question becomes, if somebody has latent TB, do you treat them to try to get rid of it? That's a good question. We're going to talk about treatment in a minute. Treatment is really difficult. So that's maybe something that's taken more on a case-by-case basis. But once it activates, what kind of early symptoms do you see? Now the patient is coughing. So a productive kind of phlegmy cough, sometimes a bloody cough. Patient has chest pain. As that progresses, you get symptoms that really consume the patient. So that's where the other name we also call tuberculosis consumption. Okay, that's where that name comes from, because the patient is really tired, they're losing weight, they're feverish, night sweats, paleness, so they're not getting as much oxygen as what they need. to their rest of their body, to the skin. That used to be really in fashion, the late 1800s. You know, poets would have this ghostly white pallor that was apparently really the thing for a while. And the other name for TB is the White Death. So that's where that name came from, because patients didn't get enough oxygen, they looked pale. Mortality about 60% at this stage if it's not treated. Okay. So the third stage, that it can get worse than this. All right, the third stage is when it's disseminated, or sometimes we say tertiary TB, where again, those macrophages are traveling all over the body and basically can infect, you know, cause a problem pretty much anywhere. So kidney function's lost, bones break, meningitis, patient can go into a coma. Okay, this is pretty much 100% fatal without treatment, and most of the time fatal even with treatment, more than 50% of the time. Okay. But it is kind of usually a slow moving disease. Okay, it's an organism that reproduces really slowly, so something that typically happens over a number of years. How do we diagnose it? Because it's such a complicated disease, that it's very complicated to diagnose it as well. So some of you may have had a tuberculin skin test. So if you've had to, you know, have that before you worked in a hospital or a nursing home, what's becoming a little bit more maybe common is a TB blood test. But one of these two is going to be the first step, either in screening you or if you have symptoms of TB, to see what's going on. Okay, so with the tuberculin skin test, you get injected with that tuberculin. That's a protein that's derived from the tuberculosis bacilli, tuberculin PPD protein derivatives, something, I'm blanking on what exactly that stands for. So you get injected with that, and then you have to come back between 2 and 3 days later, 48 and 72 hours, for somebody to read the results of that. Okay, and I was reading a little bit about this. You know, obviously, if you had some reaction like this, it's some kind of hypersensitivity reaction that they're looking for to that protein. You know, that's a pretty obvious, you know, positive. You've been exposed to this before. But there's more nuances to that. Like they'll measure, you know, the raised portion and how red it is, and take into account your age and your health status and all that, to decide what they think is going on, whether they call it a positive or not. So what does that mean if you have a positive tuberculin skin test? That means either that you've been vaccinated, which we'll talk about that in a minute, you have primary TB and you fought it off, or you have latent TB right now, or you have active TB. Okay, so this is just sort of the first stage in screening. It's one of those things. The TB blood test is a little bit nicer because they just take a blood sample and then they mix it in with some protein from the tubercle bacilli. And it's the name of this is an IGRA, so interferon gamma release assay. Okay, we talked about interferon as one of our cytokines that can interferon alpha and beta that helps us against virus infections, help guard against virus infections. Interferon gamma is actually totally different. It's released by T cells. Okay, so apparently if your T cells recognize the protein there, then they'll release this interferon gamma, so the tests if interferon gamma is released. Okay, so the nice thing is you don't have to go back and have it read 2 or 3 days later. It's a little bit more expensive, right? So it's probably not used kind of as a screening tool, but if they think you might have TB, then they're more likely to use this. And the nice thing here is that if you've been vaccinated, that doesn't cause it to be positive. Okay, so that's the first step. If you have one of those two tests and it's positive, then they will typically do a chest x-ray, right? If your chest x-ray is clear, then okay, you're clear, you don't have latent TB, you don't have active TB, you're fine. But if the chest x-ray shows up some tubercles, all right, then they have to decide, well, are these latent tubercles or is it an active case of tuberculosis? Depends on kind of how high it is in the chest x-ray. Apparently, if you have active TB, then they're usually a little bit lower in the lung. But typically, if they see any kind of tubercles, then they will proceed to the next thing and do some direct microscopy with sputum. All right, so sputum is not just what you spit out of your mouth, it's what you kind of hawk up from your lungs. All right, and then typically we'll do either acid-fast staining or some direct immunofluorescence, because we have fluorescent dyes that will recognize mycolic acid, which is kind of one of the main constituents of that acid-fast cell wall, and those will just light up really nice. So even if there's other bacteria there, you can see if there's mycobacterium there or not. And that's considered, yes, you have an active case if you have the bacilli in your lungs. So again, it's complicated to diagnose. Just briefly, how is tuberculosis passed from one person to another? And just briefly, the epidemiology here. It's only humans are the only reservoir. So with Mycobacterium tuberculosis, we pass that among ourselves. Again, there is the bovine tuberculosis that can be passed to us, but it's apparently not as serious. Okay, so we're passing it around. Transmission is considered airborne. All right, so sometimes you see that written as droplet nuclei. That just means that the bacilli can hitch a ride on really, really tiny particles that we would cough out or sneeze out, and it can stay suspended for a long time. All right, so you can be more than 6 feet away from somebody and still transmit the bacilli. Okay, so typically, again, coughing, sneezing are going to be a common transmission route. And of course, it matters how close you are to the person, what stage in infection they are, how much they're coughing out. Are you in an enclosed space or are you in a well-ventilated space? All those things matter. Again, you're predisposed if the patient has HIV. That's, again, the biggest predisposing factor. Also, as with any respiratory infection, smokers are at higher risk as well. All right, so just a couple of virulence factors. Your book doesn't really talk about any virulence factors, and when you look this up, it gets really complicated really quickly. So, and there's a lot we don't know. So we're going to mention just a few of them. That acid-fast cell wall. Again, hopefully your lab professor talked about this a little bit. And we mentioned it earlier in the semester back when we were talking about cell wall structure. The biggest component, there are other things that are unique to acid-fast cell structure, but it's that mycolic acid, which is a lipid. That's a big constituent of that cell wall structure. So you think about it, that gives the cell wall kind of a waxy consistency. All right, so when you think waxy, you think water repellent. All right, so that gives the organism a really big advantage here in terms of drying out. Okay, those mycobacterium can be coughed out of the lungs and they'll stay viable for weeks on some fomite surface. Okay, so whether that's a keyboard or, whatever, some toy, a daycare or whatever. Okay, they're also a little bit more resistant to any kind of disinfectant that you might be using to clean up around a person. But those cell walls help the organism not be likely to be digested when it's inside of a phagocytic cell like a macrophage. Okay, and as we'll see, they also have virulence factors that keep the lysosome from fusing with the phagosome. All right, so if they're not even exposed to those lysosomal enzymes, then they're going to live quite happily in there. Because of that acid-fast cell wall, they're just impermeable to a lot of antibiotics. So we have much less antibiotics that are going to be effective against this kind of infection. And I'm not sure if this is really a virulence factor, but it seemed like a good place to mention this because it takes the organism a long time to manufacture these long chain fatty acids. It grows really slowly. And that's not necessarily a bad thing for the organism. If it grows slowly, it also dies really slowly as well. So it takes us a long time to kill it. People that are on drugs for tuberculosis have to be on them for a long time, and that can be a problem. Okay, so just a couple other things to mention. This cord factor, okay, this is also a constituent of the Mycobacterium tuberculosis cell wall. And we're not really clear on what that does exactly, but we know that when we make a mutant, an organism that can't make cord factor, then that organism doesn't cause disease. So whatever that glycolipid, that molecule that's part sugar, part lipid, whatever it does, it must be important in terms of causing an infection. We know that cells that make cord factor kind of stay stuck together. So you can kind of see that in this picture here. This is not like really a great picture, but these are short rod-shaped organisms that they all seem to kind of, you know, cluster together. And you might have noticed that in lab when you looked at the Mycobacterium flea, it's almost always just big chunks of them together. Okay, I'm not sure that Mycobacterium flea actually has cord factor. I think that's unique to Mycobacterium tuberculosis. But we know that it has some role in inhibiting the migration of neutrophils. So maybe it, you know, blocks signaling that would call, you know, these immune cells to the sites. And it also plays some role in inhibiting the lysosome fusion with the phagosome. Right, so it's not exposed to all those, you know, nasty digestive enzymes. Okay, and there's other things, again, it gets a little bit beyond our scope of discussion, the actual mechanism, but we think that the organism can cause the phagosome to fail to acidify, all right, so that's going to allow it to, again, survive and keep the macrophage from becoming better at killing, keep it from becoming activated. Okay. All right, so what do we do to treat this infection? Again, streptomycin, that was an aminoglycoside that we talked about. That was the first drug that came out that was effective against TB in 1946. Of course, this was a miracle drug. They had to be very careful about how they dosed it because you get too much and that causes, you know, we talked about a kidney and ear toxicity. So, you know, life-threatening TB, it kind of warrants, you know, the trying it, even if it left the patient, you know, hard of hearing or deaf. Today, if a patient is diagnosed with kind of normal garden variety tuberculosis, they're put on a regimen of 4 antibiotics. Okay, so 2 of them we talked about before. 2 of them I thought, well, these are new, I won't worry you. But isoniazid, remember, that's the antibiotic that targets mycolic acid. So, right, very narrow spectrum. If it's only going to target Mycobacterium. Rifampicin was is a drug that targets RNA polymerase. I think that's useful against other infections, but we kind of save it for TB because we don't have a lot else that works for TB. And there's a couple others. Ethambutol is a drug that keeps the mycolic acid from inserting in the cell wall. And pyrazinamide is one that shortens the treatment time a little bit. We don't really understand why. So typically those 4 drugs, and again, don't worry about what those other 2 are called, are given to patients. Patients have to take, so this is again, when they have active TB, typically they have to be on these drugs for 6 months, sometimes 9 months, or a couple of them for 6 months, and then keep going with a couple, or all 4 of them for 6 months, keep going with a couple more for a few months. A long time. Okay, and that doesn't sound that bad, but side effects are really common. Okay, so things like nausea, headache, tiredness. Okay, those are really common things. And if patients start to feel better, their TB starts to clear after a couple months, but the drugs are making them nauseous, they're not going to want to keep taking their drugs for 6 months. So that's a problem. They stop taking their drugs, and then not all the TB is killed. It comes back, and now those drugs aren't working anymore. All right. So resistance is a huge problem in treating tuberculosis. That's why they have DOT therapy, direct observational therapy. So where people go to the homes of TB patients and watch them take their drugs and make sure that they do it. Okay, so again, it's a lot of pills for a long time that have commonly have side effects. So it's not an easy treatment. Like I said, resistance is really common. About 10% of all new cases are a multiple drug resistant strain of TB. So that means this first line of drugs, isoniazid and rifampicin, is not working. So they will try another handful of drugs. Okay, they only use streptomycin if other things don't work. So they try that, and then somewhere between 1 and 3% of all new cases are considered XDR. That means extensively drug resistant, and that means we don't have much left to treat. There was a new drug that was approved in 2012. It was the first novel mechanism to treat tuberculosis in like 50 years. And it's a drug that interferes with that mycobacterial ATP synthase. So use that in combination with some of the other drugs just to try to win out. Okay. So if treatment is hard, what about a vaccine? We do have a vaccine. Unfortunately, it's not a great one. All right. So this was developed 100 years ago, 1921, and we still don't have anything better. A couple of Frenchmen, Calmette Guerin, they named their vaccine Bacillus Calmette Guerin after themselves. So that's where the BCG name came from. This is an attenuated vaccine. So it's a strain of Mycobacterium bovis, a little milder form of TB that has been cultured kind of over and over and over in many generations, and eventually has lost some of its virulence factors that would make it cause full-blown TB. Okay, it's not terribly effective. Pretty good effectiveness in children, only about 50% in adults. Protection lasts for a time, and then you have to have another booster. And unfortunately, if you've had the vaccine, then you're going to test positive anytime you're screened for TB. So it makes diagnosis more problematic if you do end up contracting it. Okay, so because it's not all that effective and TB is not real widespread in the US, and because it makes you test positive for the skin test, we don't give it routinely in this country. But if you had somebody in your household, that you're living with somebody with TB, then that might make sense to get it. Or if you were, living for a long time in areas of the world where it's more endemic, then that might make more sense. Okay. All right, so questions about TB. Good. All right. Moving on then to chapter 20. All right. So chapter 20 is gram-negative pathogens. I typically do some gram-negative cocci and then some gram-negative rods. I'm going to leave off the gram-negative rods. We're just not going to have time for it this year. Things like E. coli. I figured the gram-negative cocci, maybe you would be a little bit less familiar because we haven't used any gram-negative cocci in lab. There's really only a couple that are pathogenic. And the ones that are pathogenic are in this genera Neisseria. Okay. All right, so again, they are going to stain red when you gram stain them. This picture from your book, they're not really very clear about whether this is a gram stain or not, but all these little dots in here, those are the Neisseria, I'm going to assume. They usually present as diplococci. And you'll see that a little bit better in a picture that I'll show you in a couple slides here. Okay, so round cells typically form in pairs. They are non-motile. So I think I mentioned when we were talking about Streptococcus that typically cocci don't have flagella. So they're typically not motile. Sometimes Neisseria are considered to have a twitching motility. Sometimes that's not really considered motility at all, but that's when it reaches out its fimbriae or pili and then uses that to hold on to something to pull it closer to it. So it's not really flagella, and it's not really like swimming around, but it's grabbing a hold of something and using it to pull it closer. Okay. Aerobic, all organisms are aerobic. Some of them are capnophiles, so they like to grow at a little bit higher concentration of carbon dioxide. They are considered fastidious. That means they're very difficult to grow in the lab. They have really picky growth requirements. You have to use a special kind of auger, special nutrients in lab in order to get them to grow. So they're hard to grow. The two most famous ones, the ones that are causing disease in humans, are Neisseria Gonorrhea, sometimes referred to as gonococcus in the literature. Also gets my vote for the hardest organism to spell. Neisseria by itself is hard to spell, and then you get all these vowels and letters that you don't hear. That's why I have that one printed out. Neisseria meningitidis, sometimes you hear that called meningococcus in the literature, same thing. All right, so we're going to look at both of those one at a time. So start with Neisseria gonorrhea. All right, so it's a causative agent of gonorrhea. How is that transmitted? You probably already know that that is a sexually transmitted disease. Okay, so that's most common way it's transmitted. It also can be transmitted mother to child. Okay, so we'll talk about that in a second. All right, so it's carried by the sperm and the body fluids, and it's going to infect different areas of the anatomy depending on the sex. So for males, it's the urethra that's going to be targeted, and females, the cervix. Okay, so because the bacteria typically kind of hitches a ride on the sperm, more common for males to give it to females. Females can give it to males, but males usually are only going to contract it about 20% of the time when they are partners with a female, where females will get it more like 50% of the time. Okay, so mother to child. So here's a picture of a child with a gonorrhea infection. So babies can be infected during the birthing process, and most often they get an infection in their eyes that can potentially lead to blindness. So babies are just treated for this, no matter whether the woman has gonorrhea or not. Some women have gonorrhea and they don't know it, or some women would lie about having it. So just all babies are treated for this. I used to put silver nitrate in the eyes as soon as babies were born. More often they'll use antibiotics drops now because that's a little bit less, irritating to babies' eyes. So just to prevent that. Virulence factors. My colleague, some of you know Dr. Baxter, he always says, I love gonorrhea because he likes to talk about the virulence factors. So if you take medical bacteriology, you will talk more about this organism and its virulence factors. We're going to kind of hit the basics here. But the big virulence factor here, typically it's referred to as pili. Your book calls it fimbriae. So I was asking Dr. Baxter about this. I was like, well, what's the catch? Anytime you read in the literature, it calls it pili, primary literature. The book calls it fimbriae. Which is it? Well, pili and fimbriae, remember, are made of the same protein. So we used to use those terms interchangeably. Now we typically, when we're talking about pili, we're talking about conjugation pili, so horizontal gene transfer. And we use fimbriae if the organism is using those structures for attachment. Well, we're gonna call them pili, or that's mostly what I'll talk about them, because for gonorrhea, all those genes that encode for these attachment structures are pil this and pil that. So we're gonna call them pili. Again, just they're not for conjugation. Okay, and their primary function is for attachment to epithelial cells in the on those mucous membranes. Okay, so there's two properties of pili. I'm getting ahead of myself maybe a little bit in terms of talking about a vaccine, but there's two reasons that you can get gonorrhea over and over and over again, and that's why we don't have a vaccine, because we don't have memory cells for it. One is those pili undergo antigenic variation. And that just means that they have a mechanism to alter their proteins in their pili. Okay, and this is not just a random mutation event, but they, again, it's a little beyond the scope of this class, but they have a mechanism to switch that around. So basically, our immune system, it takes us maybe a week or so to make antibodies, right? So we get antibodies made to those proteins on the surface of the cell. By that time, those proteins have switched, and the antibodies aren't any good anymore. All right, so then has to make up more antibodies. Oh, those aren't good, it's switched. Okay, so it's a good way for the organism to kind of outwit our immune system. Okay, so we'll say the composition of the surface proteins on the pili changes. That, in a nutshell, is antigenic variation. Okay, so it changes as soon as we get an antibody response. Okay, the other thing that Neisseria gonorrhea can do with their pili is called phase variation. And that just means they can turn certain genes on and off. Okay, so that's manifested in they are able to detach their pili. Okay, so the analogy I like is that if you are being chased by a polar bear, right, you can't outrun the polar bear, so what do? You throw down your mitten and hope the polar bear stops to sniff at your mitten, right? So they do the same thing. The antibodies, are going to attach to it. They just attach their pili. And the antibodies chase the pili, and the cell gets away. All right, so that's kind of a useful analogy, I think. All right, so other virulence factors, protease. This is an exoenzyme, an enzyme that secretes, that acts on mucosal membranes. So it cleaves that primary antibody that's found in our mucosal membranes, IgA. Polysaccharide capsule. No, does not have a capsule. Now your book says that it does. Like a lot of questions, there's some gray area there. You could actually induce Neisseria gonorrhea to produce a capsule in the lab if you grow it on a certain kind of media. But we think in the body that it does not produce a capsule. And that's almost too bad, because for Neisseria meningitidis, that organism makes a capsule and we've got a vaccine to the capsule. Okay, maybe if gonorrhea made a capsule, then we would have something that we could make a little bit more antigenic. But we think mostly not. And again, that's different from what your book says. And also lipo-oligosaccharide. Okay, that is a different form of a little different variation of LPS, lipopolysaccharide, that we found on the outer membrane of all gram-negative organisms. So essentially, this is a source of endotoxin. Okay, lipid A, part of this lipo-oligosaccharide. Basically, the sugars here are just kind of a little shorter chain than what we see in lipopolysaccharide. Okay, so it's related. All right, so remember the endotoxin that causes inflammation, fever, things like that. All right, so symptoms of gonorrhea, they're different depending on whether you're a male or female. Okay, for men, they typically burning while urinating. So I've read it's like peeing fire. All right, it's supposed to be very painful. You get pus-filled discharge there. Typically, it's painful enough that men seek treatment. Okay, so we still, for the most part, have good treatment to gonorrhea, and so we get that taken care of. So it usually for men gets treated before it gets to enough scarring that it would cause infertility. For women, it's much more common for women to have an asymptomatic infection. So it seems like, well, that's great. You know, I don't have any symptoms. It's not hurting me. Well, that's not the case. If there are symptoms, they're often mistaken for other things. So painful urination maybe can be mistaken for a bladder infection, or vaginal discharge for a yeast infection. Okay, so they're not quite as serious. The symptoms are a little bit milder, but the infection is still causing harm. Okay, it can actually, when it's not treated, because the symptoms aren't so bad, it can lead to some serious complications like pelvic inflammatory disease, which is kind of causes inflammation in the cervix and uterus, causes abdominal pain, and that can lead to ectopic pregnancies and infertility. All right, so other sexually transmitted diseases can cause symptoms like that, like chlamydia. So gonorrhea is not the only one. There is a disseminated form of gonorrhea that's getting a little bit more prevalent, where basically the organism goes systemic and cause all kinds of strange things like kind of arthritic pain and rashes. What was the other thing? Endocarditis, so inflammation of the heart muscle and heart valves. So can potentially be serious if it's not treated. Who is at risk? 18 to 24-year-olds, most common age group. So I thought that seemed like a pretty good thing to talk about. Obviously, the more sexual partners you have, the more likely you are to contract gonorrhea. It is the second most common sexually transmitted disease behind chlamydia. Okay, you can see the incidence peaked here in late 70s. That was right before the AIDS epidemic. So with you know, AIDS, public service, or you know, public education safe sex and all that, the incidence has gone down, but you can see recent years we've had an uptick. All right, and probably we're not counting all the cases. That's very likely. So, and again, we're seeing kind of an uptick in the disseminated forms. So it's not something that's going away. Okay. All right, we're out of time, so I will finish with that. I think we're still doing okay on schedule. I think hopefully we'll still get to everything next week. I'll trim back a little bit if need be, but hope you have a good weekend. Stay warm today.